is Chapter 8 for City of Species. So most of this is going to be about taxonomy and how to discern between the different domains and the different kingdoms. To start this out, I don't know if you recognize Anders Ericsson. He was on the Human Genome Project. It seems like a long time ago, relative to your lifespan probably, but not so long ago seemingly in my lifespan. It's just an interesting quote about if exceptional strain and how we use that word before expressed. So DNA gets expressed or genes get expressed into proteins. And he's just saying that, if it's eustress, not chronic stress or distress, but eustress, it can actually cause DNA or a gene to be read or expressed into a protein that normally wouldn't be. So it can actually enhance and cause you to develop in a positive way. Now, on the other hand, chronic stress could also cause it to go the opposite way and methylate DNA. And that's a fancy way of saying that DNA with epigenetics, you might have heard this, where DNA wouldn't get read, that gene would not get expressed into a protein, which could also cause negative effects. But in this quote, he's suggesting that if you challenge yourself in doable amounts, then that's how you're gonna keep increasing. You're gonna keep growing and improving. That's the potential outcome anyway. So natural selection, you've heard of this before in some realm. We're going to talk about it at microevolutional levels and how mutations set that forth. We don't know a whole lot about this. We don't know, do we know exactly what causes DNA mutations? We know some of the mechanisms, but as a very intelligent person told me at Shands, we know a lot about plumbing. We don't know much about metabolism, and mutations are part of that metabolism. And then we'll go into, well, okay, we have all these, you probably learned it in high school, maybe a mnemonic for the classifications of, the domains, the kingdoms, the phylum, the class, the order, the family, genus, species, and so on. We're going to talk about why are they in those actual classifications, and then talk about the different prokaryotes, and then go into the kingdoms themselves within the eukaryotes. This is one diagram. I'm going to have a whole section on this, but just showing you a very generic image of natural selection and showing, so the two red-faced guys, if this was bacteria, for instance, have a mutation, and then they get treated with a treatment like an antibiotic, and then whatever gene that kept them alive, that gene made a protein, kept them alive, then they pass those genes on. And we're going to talk about bacteria specifically, and then slowly but surely, you have a resistance, and then it grows and grows and grows. So I try to show a couple of different images, and you pick one that works for you, that speaks to you about the resistance, because that's how natural selection is going to be taught in this chapter. So here's the first slide on natural selection. I have two of the objectives in this chapter here. So there's a lot of small print on here. You really don't have to make it all out, but it might help if you read over it. What they're showing you in this diagram is a whole population of something. These could be termites, these could be bacteria, these could be cockroaches, it doesn't matter. The next thing is it's showing you this is a double helix, as you can see. Something happened, and a mutation of a gene, one gene, not the whole genome. Remember, those are different terms. One gene mutated. It could have been one nucleotide on the nucleic acid. See how these words just go together? So what did I use in the last 30 seconds? Genome, DNA, helix, nucleic acid, and nucleotide. And they all mean DNA. So one gene might have one nucleotide change. That's a mutation, a form of a mutation. And then if it's in the right sequence, that will make a different protein. Now, most of the time, mutations are deleterious, and that means negative. That means it's going to hurt the organism. But in this case, in this example, it's positive. And so in the image, the red guys down here in the second picture here got this mutation, so now they have a gene that makes a stronger protein. And now, if you apply, in this next picture below that, a pesticide, this is supposed to be a spray nozzle, I suppose, then you're going to spray it on this population, and who do you think is going to survive? only the ones with the mutation, because they have the genes that made the protein that allowed them to live. And then they will reproduce and then pass that to all their progeny. Now, if this was a bacteria, it could be different. They could spread their genes through conjugation and transformation, and then they could spread it through binary fission. So I'm using a lot of vocabulary here. Not all of them reproduce that way. So, and not all of them have the special power of conjugation and transformation. So it just depends. So as we go through these slides, do pay attention to that. And then once they reproduce, then you have a whole massive resistant population of pests. Again, this could be termites, cockroaches, ticks, fleas, bacteria. It doesn't matter. This applies to everything. So you have a whole population, something mutates, some of them mutate, then you apply the treatment, whatever it is for that pest, then they survive, not all of them, but just them, which actually leaves them more food, then they reproduce, and then you have a resistant population that could make the infection worse, or make the termites eating your house worse, or the flea and tick infestation worse. So think about that, play that again if you need to hear that again, but it really does apply to a lot of different pests in Florida because we're a subtropical climate. Here's another image. This is showing you the same scenario. So you can read through these of the whole population of bacteria. One was mutated, then you treat it with antibiotic, and then they conjugate and transform and or then reproduce by binary fission, making the whole population resistant. Yeah, here it's showing you, they change the colors on you, the orange, and then the whole population's resistant. This is a graph. You kind of have to stop for a second and look at it. You can see along the bottom, the x-axis are years, and then the y-axis is the different types of antibiotics. The beginning of each arrow is when they were invented, and the end of each arrow is when they became resistant. So you got to use those words carefully, because we, humans, do not become resistant to antibiotics. Bacteria become resistant to antibiotics. So you can see, you can pick one of that, like tetracycline. It wasn't around for that long a time, right? Tetracycline, it started here, and in a very short period of time became resistant. Okay, penicillin was one of the first mass-produced antibiotics, and you can see it lasted a fair amount of time. Now we still use these antibiotics, but now we have strains of bacteria that are resistant to them. And they say it's one of the biggest issues now. If you want to do any research on this, maybe for another class, they are trying to work with phages. And phages, P-H-A-G-E, are viruses that only attack bacteria. And so if you would take them in your body, they wouldn't attack you. They only attack the bacteria that they're designed to attack. So that's one angle. And also other bacteria that live in soil that might only attack other bacteria. So they are working on this. Part of the problem is there's not a lot of incentive to come up with, novel antibiotics to help combat that. It's just something to think about. Protection is a lot better than cure, because sometimes now there's not a cure. And as was mentioned earlier, every single bacterial STD is now resistant to different types of antibiotics. So people are now dying from sexually transmitted diseases that are bacterially infectious. Boy, I'm just a whole world of joy today, aren't I? Okay, so we're on to the third objective, classification of organisms. Hopefully some of this will look familiar. We have covered this in little bits and spurts throughout the class. Abiotic and biotic, you can see the root word bio there, and then you can see A in the front of one of them. So that means A means without. So in this picture, what is not living? Well, you could start with what you don't see. Well, the air is not living. Well, there's some bacteria and fungal spores in the air, but generally air is not living. Maybe the halter on the horse's head. If there's a fence back here, rocks, not living. But everything else is alive, and then you can start to classify, well, okay, ask some questions about those living organisms. What do they need? What do they eat? What can they move? What structures do they have? This is how Carl Linnaeus started the classification system way back in the day. So you have similarities of life forms. So we talked about what does it mean to be alive way back in chapter 1. Well, there's a lot of classification you could just come up with right now, not even thinking or having read the chapter. Most living organisms have to exchange gases. Most living organisms need a way to reproduce. Most living organisms have to obtain some kind of fuel source or be able to take it in somehow. So those are just some basic rules of life. Every organism that's living needs a cell. Every organism that's alive needs RNA and DNA. These are some basic reoccurring themes that are similar between all life forms. And as far as metabolic processes occurring here, well, the grass is what and the animals are what? Photosynthesizing and cellular respirating. And don't forget that photosynthesizers also cellular respirate. It's only the animals, the heterotrophs, that only can cellular respirate.
Is your business still stuck in the Stone Age with your business's printing needs? Huh? Feeling a little...
So there's a link there if you open the PowerPoint to a video if you need some more help on understanding this. We start with domain like we did all the way back in chapter 1. I know it's a flashback, right? So I found this image and I thought it was kind of helpful just because it shows the shapes. You know, you have a fungus and it's a mushroom. Actually, that's not what a fungus looks like. That's its reproductive structure. Most of the living fungus is under the ground. Animal, I don't know, is that a fish or a dolphin? Who knows? You have a leaf or a plant. This looks like some kind of weirdo blob, an amoeba, right? That's a protist. And then under this line, so these are all eukaryotes. Is that flashing back? True nucleus. Remember, they all have a true membrane-bound nucleus with DNA if it's eukaryotic. Prokaryotic means it has a prototype of a nucleus. It doesn't have a true membrane-bound nucleus, and it has a plasmid, typically, instead of, they don't call it DNA, they call it a plasmid. It's still DNA in structural form, but it's not complete, if you will. So that, and believe it or not, even though we know all the differences between these eukaryotic kingdoms, there's a lot more diversity in these two domains in the prokaryotic kingdom. Or prokaryotic domain, sorry. So it's really bizarre. Domains are more inclusive because all of these organisms, you know, I mean, the domains of the prokaryote, the domains of the eukaryote have a lot more organisms that are under their umbrella. So if we go down through taxonomy, so the classification, so I picked something that most people know, cats, okay? So just kind of like the periodic table. A lot of people think, oh, look at that thing's a mess. I can't believe I have to memorize it. You can say thank you because I didn't make you. But in science classes, sometimes they make you do that. I just want to make sure that you understand that there really is a good organizational scheme to a lot of these science diagrams and charts and tables. And again, there's a lot of organization to this one. What you have to think about is that at every level of this diagram, and we're going to start at the top and work down. Seems kind of backwards, but we're going to go from more inclusive to less inclusive. There's a question. So, what are these questions? Again, domains work from cells' needs. So, and really the kingdoms too. But domains of why are prokaryotes in here? And that's because they're just so diverse and they can use all these live in all these different environments. And eukaryotes are kind of limited. Most all eukaryotes live in the troposphere, the first layer of the atmosphere. But prokaryotes can live all over. They can live at the bottom of the ocean. They can live in different strata of the atmosphere. They can live in very inhospitable places. So let's just start with the kingdom. Okay, so now kingdoms are plant, animal, fungi, and protist, right? So the first question is, what do the cells need? Well, in this kingdom, you're going to look at these different kingdoms and you're going to say, well, okay, fungus, they don't photosynthesize. They're more like a actual plant than they are an animal. I mean, more like an animal than a plant. Sorry about that. Because they don't photosynthesize. Plants photosynthesize and get their own energy for their cells. Protists have to live in water, and they have to engulf their food, and they're single celled, but they do have to go get their own energy. And then, animals, how they get their own energy, they're heterotrophs as well. So the kingdoms are actually kind of looking at what the cells need for energy. And then you go into the phylum, and you see chordata, and I think of this as spinal chordata. It has a backbone. It has vertebra. It has discs in between the vertebra. So then the cat would now move up into chordata. Well, then you go up and you see mammal. Well, I think most people know mammals. So mammals, mammary glands, or mammary glands, or breasts. It means that the animal will give milk to its young. Now, there are some weirdo exceptions. We're not talking about exceptions. You can think of, go into marsupials and you'll lose your mind. But mammals, for the most part, they have to give milk to their young. That's why they're in that category. And these categories morph. That's why I don't make you memorize them. That's why I hit you from the angle that, why did they put them here? What questions were the scientists asking? And then you move up a little bit further and you see carnivore. Well, you know what question that's asking. It's starting to talk about energy needs. Well, specifically, cats are carnivore. They don't eat plants. Now, dogs are omnivores, and they'll eat, pigs are omnivores. They'll eat plants or animals. And all you got to do, most of you might know this, just look at an animal's teeth. If an animal only has sharp, pointy teeth, they're carnivores for the most part. I never say never in science. It's like, well, there's never always, it's never, never. It's according to what we know now. And then, so they're going to eat meat, and it's usually based on dentition and the intestinal tract. What kind of bacteria and organisms live symbiotically with them to help them use their food and their nutrients. And then you go up a step further to, more specifically, instead of canine, this is where dogs and cats would separate, you go to the feline. And then you go even further and you talk about retractable claws or not retractable claws, and so on and so forth. So it's not about a lot of memorization. It's understanding that there's a question being asked. It's kind of like when you clean your room and you have drawers and boxes and areas, and you say, well, where would I go to find this again? And then you put it in that area. It's not exactly like that, obviously, but similar to that. So that's how the classification system works. And maybe if you look at it differently now, it won't look so annoying and science-y, and it'll actually make a little bit more sense to you. So objective number 4, compare and contrast bacteria, protists, fungus, and plants. So we are going to look at bacteria first because they're in their own domain. There are 2 domains of bacteria, archaea and eubacteria. For the scope of this class, we're going to talk about bacteria because we're going to talk about the bacteria that typically interact with humans. And I use that word loosely. We're going to talk about that. Then we're going to go into the eukaryotes, which are protists, fungi, and plants. Most animal, we're going to refrain, and we're going to look at that in chapter 9 as an individual chapter for animals. So you might want to pause it. I'm going to do a spoiler alert for these pictures below. I will give a caveat if you're going to try to guess these, because I don't want to frustrate you. The magnification should probably be put on there to help you. Some of them are naked eye, and then others are 1,000 magnification. So maybe I can help you out. This one here, this spiral, is 1,000 magnification. This is about 100 magnification, naked eye, 400 magnification, naked eye, down here, naked eye, naked eye, naked eye, and this gold discs are about 1,000 magnification. So that might kind of give it away too if you know anything about microscopy. So I'm going to start from the top left. This is ringworm. Not an animal, not a worm. It's actually a fungus. Next to it, on the right, is sleeping sickness. It's a protist, and these are red blood cells. So this is a protist swimming around in someone's blood. Yay. Delivered by a fly. These are mold spores about to be released from a fungus reproducing. This is a piece of cheese which can be flavored by both mold or bacteria. So cheese, obviously it's made from milk product from cows or goats or different mammals, but the flavoring, the different flavors of the cheeses is due to fungus or prokaryotes. Down here on the left is slime mold. That usually goes in the produce kingdom, but I have seen that flip back and forth, and I would never ask you that, even though it does look like a fungus. And this is a prokaryote. This is a spiral bacteria. I forget the actual species of that. And this is a fungus. This is the reproductive structure of a fungus. And this is a slime mold growing on some kind of fruit. So that is a protist actually, because it's slime mold. Even though it's a mold, it's in the protist kingdom. And then here above that, right here, the gold disc is actually MRSA. Looks kind of pretty to be MRSA. Looks like grapes. But it's methicillin-resistant Staphylococcus aureus. So I'm going to post these questions and give you a second to look at them and try to answer them. So you might want to pause the video, because I'm going to go ahead and give them away, just in case you want to see if you're right. So the first one's, all toxins can be destroyed by cooking sufficiently. No, because some toxins are not destroyed by heat or pressure. So even a pressure cooker wouldn't get rid of them. Steak lasts just as long as hamburger. No, and hopefully you tried to answer which one would last longer and why. Hamburger's ground, so it's exposed to the air, so it's exposed to more pathogens, so it doesn't last as long. Steak is a muscle meat, so it's sealed on the inside. The only thing exposed to the air is the outside, and when you cook that at high heat, it usually kills off the foodborne pathogens and can last up to 14 days in the refrigerator, even uncooked if it's wrapped right. Thawing meat in the sink and the counter is fine. For most people, maybe, but would you want to take the chance if you're immunocompromised and don't know it? Because once that meat gets to room temperature, the bacteria start reproducing. And you might think, well, it's still frozen in the middle. Yeah, but that still allows the outer portion of the meat to grow to infectious levels of a foodborne illness. And that's where you're going to get in trouble. Because if it gets too high and it's inside the meat, the heat might not reach it to kill it. And there's your issue. The best thing to do is plan ahead, I know, and put it in the refrigerator and let it thaw there and then cook it. Cooking times, temperatures for meat are specific to each type. Yes, look that up and use a good thermometer that's calibrated and works. Because especially if you're a hunter, because if you're going to hunt wild game, they are more susceptible to pathogens. I agree they'd be healthier because of less hormones and they're out eating natural food. I'm not putting down that. I'm just saying they could have pathogens that you're not used to because they're wild, and you want to make sure you cook that meat to temperature, especially wild boar. Swine carries a lot of bizarre pathogens, and you definitely want to cook that meat to the proper temperature and time. Door of the refrigerator is a good place to store milk. No, not anything that perishes fast, because the temperature changes rapidly as it's opened and might be left open and not realize it. Food that's a foodborne pathogen smells and looks fine. No. Sorry, yes, that's true. It could look and smell fine. It could be right on the verge, and some don't give off smells and might not appear from any visual aspect as being at infectious level. FIFO, first in, first out. Yes, you should write dates on canned goods so that you know you're using the oldest one first. So especially in Florida with storms, you don't want to be going and grabbing a can that's bloated and then you wind up getting botulinum. That would not be good. And then foodborne illnesses, the only caused by bacteria. No, you're going to learn soon that it can be caused by a lot of different pathogens. And then down here on the right, you have the now 20-second rule. It's good for your immune system, right? And so we're in the domain prokaryote. And this is conjugation and transformation, a picture up on the right. So the bacteria on the top has a gene that, let's say in this case, makes this resistant to an antibiotic treatment. What it's doing, literally, this is an electron microscope image, growing a pili, which is Latin for hair, to the other bacteria that doesn't have the gene, and it's going to send the gene through that tunnel to this one, and now he is going to be resistant to the antibiotic, and then they're both going to go through binary fission. So let's walk through that one more time. This one has the gene that makes it resistant to the antibiotic, and so it is already transformed because it has the gene. Then it sends the gene to the other bacteria. That's a conjugation. Conjugation means to come together. And now this bacteria, once it gets it, is transformed. So conjugation, transformation, and then both of these guys will binary fission. They'll go through binary fission, and then they will both exponentially reproduce, and sooner or later that can become a really bad resistant infection. This is another diagram of it if you need something more technical. This one with all the little strings coming out of it, that would be the one here, the donor. And then this one down here is the recipient. You can go through those steps. And then I have a GIF on it, just, I don't know, kind of goofy, but sometimes it helps you to see it. Right here is the conjugation with the tube forming, and then the gene is sent, and then it becomes transformed. To be or not to be, that's funny. And then you have the resistant bacteria down here in the little cartoon on the bottom. So here is binary fission. You can kind of see these Salmonella bacteria. This is probably a very good 1000 magnified image of Salmonella going through binary fission. See this little cleavage furrow in the middle? So they're just about ready to become two bacteria. And here is what it looks like magnified about 10,000 times. So this is not conjugation transformation. Notice this is very close together. What they did was blow up that little cleavage there. So you can see this is just like mitosis in us. So binary fission is like mitosis. We do go through asexual reproduction. So binary fission is like mitosis, which is a form of asexual reproduction, but we don't produce more of us, we just produce more of ourselves. Be careful with that. Not ourselves, cells. So these guys are actually, because they're single celled, they're reproducing more of each other. And some questions to go through. Hopefully you've answered that one. No. We need a ton of bacteria living symbiotically in us to keep us healthy. We're going to go into that. I covered a plasmid in the last slide. This is technically its DNA, and technically once it's set up in each bacteria separately, it'll become a plasmid. Producer, consumer, decomposer. So from another chapter we talked about this. Producer means photosynthesizer. Consumer means they have to get a carbon source from another area, so a heterotroph. And decomposer means they can feed off of dead things. And let me be very specific on that. Feed off of dead things and return molecules to an elemental form. That's very, very important to the environment. If we do not have organisms that take molecules and break them down to elements, then
We tried everything, but Charlie still struggled with math. Then another month.
Nutrient cycles won't happen, that we're going to cover in chapter 10. So yes, bacteria are all three of these, and you do not have to know individual examples. Mutualistic, commensalistic, or parasitic? Yes. Short answer. Mutualistic, live together with a host, absolutely, because without bacteria in and on us, we would not be alive. We could not make our own nutrients, neurotransmitters, brain-derived neurotropic factor, a whole bunch of things. Commensalistic, they live on something and are benefited but don't harm the host. Yes, we have some of those in and on us. Parasitic, we have some of those in and on us too. So you could apply bacteria to all three of those on a human. So they do each one of those ecological symbiotic relationships. And we covered that. So binary fission is like mitosis, asexual, and then conjugation and transformation is very specific to sending different genes to each other. How are bacteria used for humans? We're going to go into that with the microbiome. And Botox, MRSA, tetanus are all examples of pathogenic bacteria. Now, Botox, you could say, well, no, Botox is good for you. It makes your skin look less old. Well, right, if it's used right. It can also kill you, which makes it kind of scary because you're injecting it into your head. But you're supposed to have very skilled people do that, because if they were to put too much, it could cause serious complications. Because what the botulinum toxin that they use to paralyze your face muscles so you don't have wrinkles is exactly that. It paralyzes muscles, so you could see very quickly how that could go south. And now we're going to go into the microbiome, and we're going to really go into that more in chapter 9, but there is a lot to pick up on this, and I've kind of in dribs and drabs covered it because science is kind of picking up on it in a big degree right now. Over the last 10 years, the research has blown up on this, and not only on the microbiome, but how we've overlooked it and how it has so many other ties to our health, and also diseases that we used to think were caused different ways. So it's really something to read about. So there's links on these PowerPoints for a lot of things. There's a video on this. There's a link to the NIH page about the Human Microbiome Project. We're going to specifically cover this from the gut microbiome. There are microbiomes all over you. They're on your skin, they're in your mouth, they're in your armpits, your groin, between your toes, your ears, but we're specifically going to look at it in the large intestine. That's where the major part of it lives, and the major part of health being kind of simultaneously created for us is having a big impact. So what does it do for us? This is a diagram that just gives you a very superficial look. I know you're thinking, that's a lot. Well, there's a lot in the book on this too, but more specifically, it's the colon or the large intestine that's the issue. Now, don't ignore the other parts of that diagram, but the large intestine is where your diet specifically, because think about it, okay, if we need bacteria in our large intestine, because our bacteria that are living symbiotically, mutualistically, we're feeding them our food, and in exchange they make vitamins and minerals and neurotransmitters, believe it or not. 90% of our serotonin is made in our large intestine. I know, it blows my mind. No pun intended, because serotonin we used to think was only made in your brain. It also makes hormones, short-chain fatty acids that make all sorts of things for us. So without that, so let's just say you all of a sudden start eating high ultra-processed food, a lot of sugar. I know I'm back on my soapbox, but you're going to kill off your microbiome, because your microbiome doesn't live on high-processed food. Your microbiome lives on fiber. I know. Wah, wah, wah. It can live on other things, but the one thing they know it doesn't live on is high processed food and sugar. It's people that go on the carnivore diet, they do well for bits and spurts of time, and they're still working on this. They're not. Some people are forming conclusions. I think it's very, very. It's too soon to form conclusions on this. I just think it's amazing what they're learning about it, and I hope they keep an open mind. But just changing your diet in 2 weeks can turn your life around. Depression, they're now linking to the microbiome of the gut. They're linking all these autoimmune disorders. They're linking celiac. We used to think that celiac was caused because of a genetic link. Well, now we're thinking, wait a second, maybe it's a poor diet or something you're missing from your diet that's not feeding the bacteria, and now you have celiac. Maybe it's the other way around. So if you're suffering from some kind of bizarre issue, it can be anything from depression or whatever, try it. Try eating a really clean diet with a lot of plant fiber. Humor yourself, right? Give yourself 2 weeks to a month. Cut out the sugar and just see if it makes a difference. And not eating too late at night is another thing. It's turned people's lives around. There's a ton of testimonies about this on YouTube from legitimate sources, not from fly-by-night people. So gut flora. So you probably heard flora and fauna. Fauna means animals. I think a baby fawn, like a deer. Fauna, but it's not spelled that way. It's F-A-U-N-A. Flora means photosynthesizers. I don't know why they call it that, because they're animal-like, not plant-like, but we'll go with it. Gut flora means the microbes that live in your gut. And so the two terms that might come to your mind are prebiotics and probiotics. prebiotics are what you feed the probiotics. So prebiotics are fiber. You know, you don't bring home the probiotic, you don't bring home the horse before you bring home the hay. You might be laughing and thinking, oh, I bring them home at the same time. But anyway, the prebiotic is the fiber, and the probiotic are the bacteria that give you health. And you don't have to go and buy anything as a supplement. You can save yourself a ton of money. You want to slowly break into this, start drinking unpasteurized milk if you're capable. If you're immunocompromised, I would not do that. But find a local farm where you can go to and see it be milked. You want to make sure they're not just buying it off the shelf, because that's not helpful. And drink that. That will give you a lot of natural probiotics. And then prebiotics are fibers and whole foods from plants. So you eat those simultaneously, and over a couple of weeks, you're going to repair if you damaged your microbiome in your gut. 2 weeks tops should do the trick on that if you keep to it and do a little bit each day. But yeah, they want to sell you a lot of stuff. One of the problems is if you only buy probiotics and you're not eating fiber, you're really just going to the bathroom and wasting it, so it's not real helpful. Things to enhance it, yes, I just covered those with whole foods, fiber, harmit, sugars, ultra-processed foods. And they say that, in a couple of these video links, if you lose your microbiome, you wipe it out with junk food. The video link is really good. It's about the analogous to losing your liver, and you can't lose your liver. I mean, that's, that gets something, it's an alchemist, it detoxifies, it creates a lot of other molecules in your body and cleans your blood. It's pretty impressive. So what they're saying is it's pretty serious. Here's another diagram. They call it another organ, the invisible organ system. It's like another organ in your body, quite literally, and how it operates to keep you healthy. Fecal microbiome transplant. So that's a very interesting thing, and I'm not going to go into it, but if you want to look it up, because they've done a lot of research on this, and they can turn people's health around within 24 hours. And they're doing this at Johns Hopkins and Mayo Clinic. It's not some quack shop setup. So fecal microbiome transplants, FMT, and it has a phenomenal amount of benefits to it. There are some risks, so you would have to really want one, like for a health issue, like serious health issue, but the benefits it really surprised a lot of people. And relating this to chapter 2 with the glycocalyx, how a really poor diet can wipe that out in your vascular system, so can a poor diet wipe out the microbiome and intestinal lining. And what they're showing in a lot of the research is that, quite literally, you have a mucous membrane that protects your intestinal lining, and when you eat that junk food, that high ultra-processed food, it wipes out that mucous lining, and that's where all your microbiome lives. And once that's gone, you can get leaky gut. And leaky gut anymore is not a new age kind of naturopath issue. It's embraced by Western medicine, and they're linking it to a lot of very novel immunocompromised situations and autoimmune disorders, actually. That's one of the reasons leaky gut is why they're saying that a lot of cases of celiac aren't genetic. They're actually caused by problematic eating. And we will definitely revisit prokaryotes in chapter 10 because they are the basic reason for the nutrient cycles, such as the carbon cycle, the nitrogen cycle, the sulfur cycle, and the phosphorus cycle.